Basics of Medical Biochemistry. First edition, 2023. Chapter 17. Chemistry of Nucleic Acids, DNA Replication, DNA Damage and Repair. Part 3 of 3. By Professor Dr. M.Jawed. MBBS, Tao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College, Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 17. Chemistry of Nucleic Acids, DNA Replication, DNA Damage and Repair. 17.8 DNA Damage. In human cells, both normal metabolic activities such as replication and environmental factors such as ultraviolet, UV, light and radiation can cause DNA damage. Inside the cell, DNA damage occurs at the rate of 1000 to 1000,000 molecular lesions per cell per day. Many of these lesions cause structural damage to the DNA molecule and can alter or eliminate the cell's ability to transcribe the gene that the affected DNA encodes. DNA damage results in physical or chemical changes in its structure. This may block replication, alter transcription, or generate mutations. DNA damage can be subdivided into two main types: one, exogenous causes as by external agents such as ultraviolet, UV 200 to 300 nanometers, radiation from the sun, other radiation frequencies including X-rays and gamma rays, human-made mutagenic chemicals especially aromatic compounds that act as DNA intercalating agents, cancer chemotherapy and radiotherapy. Two, endogenous causes such as attack by reactive oxygen species produced from normal metabolic byproducts, spontaneous mutation, especially the process of oxidative deamination. Endogenous causes also include replication errors. The following are the main types of damage to DNA due to endogenous cellular processes. I. Oxidation of bases, example 8-oxo-7,8-dihydroguanine, 8-oxo-G, and generation of DNA strand interruptions from reactive oxygen species. 2. Alkylation of bases, usually methylation, such as the formation of 7-methyl-guanine, 1-methyladenine, and O6-methyl-guanine. 3. Hydrolysis of bases such as deamination, depurination, and dipyrimidination. 4. Bulky adduct formation, i.e., benzoapyrenediol epoxide DG adduct. V. Mismatch of bases due to errors in DNA replication, in which the wrong DNA base is stitched into place in a newly forming DNA strand, or a DNA base is skipped over or mistakenly inserted. DNA damage and mutation are fundamentally different. Damage is a physical abnormality in the DNA, such as single and double strand breaks, 8-hydroxydioxyguanosine residues, and polycyclic aromatic hydrocarbon adducts. In contrast to DNA damage, a mutation is a change in the base sequence of the DNA. A mutation cannot be recognized by enzymes once the base change is present in both DNA strands, and thus a mutation cannot be repaired. At the cellular level, mutations can cause alterations in protein function and regulation. Mutations are replicated when the cell replicates. In a population of cells, mutant cells will increase or decrease in frequency according to the effects of the mutation on the ability of the cell to survive and reproduce. 17.8.1 DNA damage by base loss. The glycosyl bond linking DNA bases with deoxyribose is labeled under physiological conditions. Within a typical mammalian cell, several thousand purines and several hundred pyrimidines are spontaneously lost per diploid genome per day. Loss of a purine or pyrimidine base creates an apurinic/apyrimidinic (AP) site, also called an abasic site. Figure 17.22 Appearance of a purinic/apyrimidinic (AP) site as a result of a base loss. 17.8.2 DNA damage by base modification, deamination. The primary amino groups of nucleic acid bases are somewhat unstable. The amino group is removed as ammonia. For example, in spontaneous deamination of cytosine, there is the hydrolysis reaction of cytosine into uracil with the release of ammonia. Cytosine plus H2O uracil plus NH3. In a typical mammalian cell, about 100 uracils are generated per haploid genome per day in this fashion. Other deamination reactions include the conversion of adenine to hypoxanthine, guanine to xanthine, and 5-methylcytosine to thymine. 2. Chemical modification. The nucleic acid bases are susceptible to numerous modifications by a wide variety of chemical agents. For example, several types of hyperreactive oxygen, singlet oxygen, peroxide radicals, hydrogen peroxide, and hydroxyl radicals, are generated as byproducts during normal oxidative metabolism and also by ionizing radiations such as X-rays, gamma rays, etc. These are frequently called reactive oxygen species, ROS. ROS can modify DNA bases. A common product of thymine oxidation is thymine glycol. Figure 17.23 Formation of thymine glycol by oxidation of thymine. 17.8.3 Photodamage. Ultraviolet light is absorbed by the nucleic acid bases, and the resulting influx of energy can induce chemical changes. The most frequent photoproducts are the consequences of bond formation between adjacent pyrimidines within one strand, and of these, the most frequent are cyclobutane pyrimidine dimers, CPDs. Ultraviolet light induces the formation of covalent linkages by reactions localized on the C=C double bonds. 17.8.4 Interstrand cross-links, by attaching an agent to bases on both strands. For example, a bifunctional alkylating agent such as the psoralens can cross-link both strands. Cross-links can also be generated by UV and ionizing radiation. 17.8.5 Strand breaks. Single-strand and double-strand breaks are produced at low frequency during normal DNA metabolism by topoisomerases, nucleases, the collapse of the replication fork, and repair processes. Breaks are also produced by ionizing radiation. 17.9 DNA repair. It refers to a collection of processes by which a cell identifies and corrects damage to the DNA molecules that encode its genome. All cells are capable of restoring the damaged DNA to its original form. Most of the enzymes involved in repair can recognize the lesion, remove the damaged section of the strand and fill the gap by using the sister strand as a template. If damaged DNA is left unrepaired, the consequences are lethal to the cell, which may cause mutations that may result in abnormal cell growth. The following are the important mechanisms of repair. 17.9.1 Direct repair. DR cells are known to eliminate damage to their DNA by chemically reversing it. These mechanisms do not require a template, since the types of damage they counteract can only occur in one of the four bases. Such direct reversal mechanisms are specific to the type of damage incurred and do not involve breakage of the phosphodiester backbone. A damaged base undergoes a chemical/UV reaction to restore its original structure, example DNA photolyase in E. coli. 17.9.2 Base excision repair. It is a major pathway for the repair of modified bases, uracil misincorporation and oxidative damage. Various DNA glycosylases recognize lesions and remove base at glycosidic bond, thereby producing an abasic or AP, a purinic/apyrimidinic, site by base flipping out. One of several AP endonucleases incises phosphodiester backbone adjacent to the AP site. AP nucleotide is removed by an exonuclease and the patch is refilled by DNA synthesis and ligation. Figure 17.24 Basic mechanism of base excision repair, BER. 17.9.3 Nucleotide excision repair, NA. It targets large, bulky lesions and removes DNA on either side of them. In contrast to base excision repair, NA does not require specific recognition of a damaged nucleotide and thus it can remove DNA lesions. Figure 17.25 Basic mechanism of nucleotide excision repair, NA. Xeroderma pigmentosum, XP, is an autosomal recessive genetic disease. The clinical syndrome includes marked sensitivity to sunlight, ultraviolet, formation of a blister on the skin, and dry and hyperpigmented skin. Corneal ulcers may be present. The risk of developing skin cancer is increased by 1,000 to 2,000-fold. The inherited defect seems to involve the repair of damaged DNA, particularly thymine dimers. Cells cultured from patients with xeroderma pigmentosum exhibit low activity for the nucleotide excision repair process. The prognosis is poor and usually results in premature death. 17.9.4 Mismatch repair, MMR. It corrects the errors of DNA replication and recombination that result in mispaired, but undamaged, nucleotides. It is carried out by following 3 steps. I. Recognition of the mismatched nucleotide. 2. Removal of incorrect nucleotide. 3. Addition of correct nucleotide by DNA polymerase III and then joining of the strand by DNA ligase. Mutation of the proteins involved in MMR in humans is associated with non-polyposis colorectal cancer, Lynch syndrome. Figure 17.26 Basic mechanism of mismatch repair, MMR. 17.95 Repairing of strand breaks. Ionizing radiation and certain chemicals can produce both single-strand breaks, SSBs, and double-strand breaks, DSBs, in the DNA backbone. Single-strand breaks, SSB, repair. Breaks in a single strand of the DNA molecule are repaired using the same enzyme systems that are used in base excision repair, BR. 2. Double-strand break, DSB, repair. There are 2 mechanisms by which the cell attempts to repair a complete break in a DNA molecule. A. Direct joining of the broken ends. This requires proteins that recognize and bind to the exposed ends and bring them together for ligating. This type of joining is also called non-homologous end joining, NHEJ. A protein called Cu is essential for NHEJ. This method of repair is mutagenic because it removes several base pairs at the break site mediated by Cu proteins. Errors in direct joining may be a cause of the various translocations that are associated with cancers. Examples include Burkitt's lymphoma, Philadelphia chromosome in chronic myelogenous leukemia, CML, B-cell leukemia, etc. B. Homologous recombination. Here the broken ends are repaired using enzymes that normally perform genetic recombination between the homologous chromosomes during meiosis. This system is much less error-prone than non-homologous end joining. 2 of the proteins used in homologous recombination are encoded by the genes BRCA1 and BRCA2. Inherited mutations in these genes predispose women to breast and ovarian cancers. Figure 17.27 Basic mechanism of double-strand break repair. Summary. A DNA molecule is composed of 2 polynucleotide strands, double strands, that spiral around to form a double helix, while an RNA molecule is a single polynucleotide chain, single strand. Each nucleotide is composed of a nitrogenous base purine or pyrimidine. 2-pentose sugar is deoxyribose in DNA and ribose in RNA. 3-phosphate group. A nucleoside is formed by the combination of a purine or pyrimidine nitrogenous base with pentose sugar, ribose or deoxyribose. Purines include adenine, A, i.e. 6-aminopurine, and guanine, G, i.e. 2-amino-6-oxypurine, both of which are found in DNA and RNA. Pyrimidines include cytosine, C, thymine, T, and uracil, U. Cytosine is found both in DNA and RNA, whereas thymine is found only in DNA and uracil is found only in RNA. The base sequence of the polynucleotide is always read and write from the 5-to-3 direction. Example 5 GCAT3. Adenine on one strand binds to thymine, A equals T, of the opposite strand by 2 hydrogen bonds. Guanine on one strand binds to cytosine, GC, of the opposite strand by 3 hydrogen bonds, which is called base pairing. One strand in DNA is a coding strand and the other is the template. The double-stranded DNA wraps around a basic protein, histone octamer core to form the nucleosome. Segments of DNA specifying the synthesis of a functional protein or RNA product are called genes. The coding part of the gene is known as an exon, whereas the non-coding region is termed an intron. The term junk DNA refers to the non-coding regions of DNA. It has no apparent function. The main function of DNA is to store, express and transmit genetic information. It can also undergo mutation, which is essential for the evolution of new varieties and species. RNA is mainly concerned with the synthesis of proteins. There are three main types of RNA: 1 messenger RNA or mRNA, 5 to 10%, 2 transfer RNA or tRNA, 10 to 15%, and 3 ribosomal RNA or rRNA, 75 to 80%. mRNA act as a messenger for transferring genetic information from the gene of DNA to the ribosomes. Function of tRNA is to carry amino acids to the site of protein synthesis in ribosomes, while rRNA takes part in the formation of ribosomes and binding of mRNA to ribosomes. Codon/code is a sequence of 3 successive nitrogenous bases in the molecule of mRNA. Codons are regarded as the key to the translation of genetic information during the synthesis of specific proteins. Replication of DNA is the process of duplicating, reproducing, or doubling DNA molecules. It is the biosynthesis of an exact copy of DNA molecule from monomeric deoxyribonucleotides. Replication of DNA includes 3 major steps: 1 initiation, 2 elongation and 3 termination. Leading strand, the strand of DNA which is copied in the direction of the advancing replication fork. It is synthesized continuously, from outside of the fork to the origin. Lagging strand, the strand of DNA which is copied in the direction opposite to the movement of the replication fork. Telomere is a repeated Tg-rich hexanucleotide sequence located at the end of each chromosome. Both normal metabolic activities such as replication and environmental factors such as ultraviolet, UV, light and radiation can cause DNA damage. Exogenous causes of DNA damage include external agents such as ultraviolet, UV 200 to 300 nanometers, radiation, X-rays, gamma rays, human-made mutagenic chemicals especially aromatic compounds, etc. Endogenous causes include attacks by reactive oxygen species produced from normal metabolic byproducts, spontaneous mutation, especially the process of oxidative deamination and replication errors. Loss of a purine or pyrimidine base creates an apurinic/apyrimidinic AP, site, also called an abasic site. Spontaneous deamination of cytosine can convert it into uracil with the release of ammonia. DNA repair refers to a collection of processes by which a cell identifies and corrects damage to the DNA molecules. Xeroderma pigmentosum is an autosomal recessive genetic disease. The patient has a marked sensitivity to sunlight. This disease is due to low activity for the nucleotide excision repair process. References. Exercise questions. For MCQ 1 to 4 see Part 1 and for MCQ 5 to 8 see Part 2 of this chapter. MCQ 9. A 70-year-old man suffering from lung cancer was admitted to the critical care unit for complaints of a typical catechetic condition. A thorough investigation revealed that along with many other causes, he is also deficient in specific RNA concerned to kill cancer cells. Which of the following types of RNA is deficient in this patient? Heterogeneous nuclear RNA, hnRNA. MicroRNA, mRNA. Ribosomal RNA, rRNA. Small interfering RNA, siRNA. Transfer RNA, tRNA. MCQ 10. A 1-month-old boy was brought to the hospital by his parents for complaints of some abnormality in the shape of his ears. He suspected some genetic problem. A series of genetic tests were performed during which, the base sequence on a segment of one strand of DNA was detected as CAG-TAG. Which of the following base sequences will be on the corresponding segment of DNA under normal circumstances? CTACT, CTATK, GOTK, TAGCAGT, TAGCAT. MCQ 11. Which of the following is a key function of DNA repair mechanisms in cells? DNA damage correction, DNA mutation, DNA replication, DNA transcription. MCQ 12. Mutations can arise from various sources. Which of the following is a major contributor to spontaneous mutations? Chemical mutagens, replication errors, ultraviolet, UV, radiation, X-ray exposure. End of Part 3 of 3 of Chapter 17. Thank you.